So first of all, hello everyone who has joined us for this session this week. I just want to welcome those who have joined us for the previous sessions, and thank you for joining us once again for this Malaysian Research Insider session. And for those who are new, welcome as well. I hope you'll enjoy our session today. My name is Alia Marzuki, and I will be hosting today's MBIOS Researcher Insider session. So this science chat today is co-organized by MBIOS, 100 Scientists of Malaysia, along with our media science partner, Science Media Center Malaysia. Sorry, so Sara is doing a PhD in neuropharmacology in the University of Nottingham, UK. Her project explores intranasal delivery of oxytocin for neurodevelopment disorders. And she also is a co-founder of the 100 Scientists of Malaysia project, which aims to highlight Malaysian researchers working in STEM. And their Facebook page currently has over 6,000 followers. So that's super, super impressive. So I'll now leave the floor to Sara to begin her presentation.
Hi guys, sorry, let me just share my screen. Cool, can everyone see my screen with my slides?
Yep, can see it.
Okay, cool. So thanks MBIOS for inviting me, Apple especially for asking me to come on to Research Insider. I'm going to spend about 15 minutes, hopefully not over 20. The first two slides will just really be who I am, where I'm from, and then I'll go into my main play of my research, which is oxytocin. So what is oxytocin and why do I use oxytocin? And once that's done, I'm going to go into more into my research techniques on what I do to answer my research questions. So, sorry. Yeah, so I did my entire education in JB, where I'm from. So that's 15-year-old me, convent, SPM in JB. And then I went on to do STPM in JB as well. I think I'm somewhere at the back. And then I went on to do my biomedical science degree in King's College London, where I focus on more neuroscience and pharmacology towards my final year modules. Currently, I am a third-year PhD neuroscience student at Nottingham University, funded by the Vice Chancellor Scholarship. And my project, like Alea mentioned, is really to improve delivery of oxytocin to the brain via intranasal application, which is basically putting it up the nose to reach the brain better. So before I move on to oxytocin, I think I have just one poll up for people, because I'm not sure if many of you have heard of oxytocin. You might have, but I was just curious to know how much you know about oxytocin. So yes, all of above, that is accurate. So oxytocin is a hormone and a neuropeptide produced naturally in the brain by something called the hypothalamus. So it's this brain structure at the base of your brain. And the hypothalamus really is responsible for regulating how your body works. So like homeostasis, for example, regulating your appetite and that sort of thing. So it secretes hormones, for example, growth hormones, leptin that regulates the appetite, as well as oxytocin. So you might have heard of oxytocin being dubbed as the love hormone by the media. And that really is because they pinpoint on the fact that oxytocin is released during social interaction, when you cuddle with your significant other, when you pet your dog, all of those actions release oxytocin. So it's a feel-good hormone, really. But besides that, oxytocin's main effects really is in childbirth and breastfeeding. So for example, during labor, nurses and doctors can administer something called Pitocin, which is a man-made version of oxytocin to stimulate your contractions of your smooth muscles so that childbirth can happen. And then it's also via suckling at the nipple can stimulate milk letdown for breastfeeding. Oxytocin is also found, the receptors of oxytocin is also found in a lot of other different parts of the body. So for example, your heart, your gastrointestinal tract, your kidneys and all that. So it does have a variety of effects across your body, and it's not specifically just the love hormone, as a lot of you might have heard of it. But so the oxytocin hype cycle, this really comes about, so we all knew about its effects on the childbirth and lactation, but then when it came to social behavior, it really began to hype in the early 1970s, where they did experiments in animals. So for example, rats, rats, unlike humans, where say, for example, you might see a really cute baby on the street and you'd be like, oh, I really want to cuddle that baby or like protect it from harm and all that. But rats are not the same. So unless if it doesn't have pups, it has no interest in like stranger pups, and you do nothing towards it. So what they did was they injected rats, virgin rats, with oxytocin, and found that the virgin rats adopted some maternal behavior towards pups that are not her or her own. So that was really one of the first experiments, and then a few more experiments came up and replicated that sort of effect. Another rodent mammal that's used in, sorry, the picture didn't come out. So another rodent mammal that is used in this sort of experiment is called the prairie vole. So the prairie vole are special because they mate for life. So what they found was that when they injected oxytocin into a prairie vole, it induced pair bond formation with another prairie vole, and then by blocking that receptor, it abolished the effects. That's a prairie vole. It didn't come out just now. And then in 2005, there was a really big study saying that oxytocin increased trust in humans. So basically, what happened in that study was that as an investor, you would have to trust in someone else by giving them your money and trust that they would kind of reciprocate the effect back and give you and share that money with you, or they could just take the whole bulk of it and leave you with nothing. So by giving oxytocin to the investor, they found that it actually increased trust in them and made them more willing to kind of share that money and believe that they would get something back from the other person. Then there's also many, and then it was followed by many experiments. For example, if you knock out oxytocin in a lot of animals, it really showed it played a role in social recognition, so recognizing someone else, social interaction, and as well as cognition and memory. So that blue bit, that really was the hype of oxytocin and excited a lot of researchers in using them for diseases such as autism and schizophrenia, where these symptom domains that I mentioned currently don't really have any treatments for them. But with every up, there's always a down. And then that was when things started to begin like, oh, maybe oxytocin is not that great. So it was shown that it increased negative effects, for example, envy and gloating. It increased violence inclination amongst people who already had traits towards aggressiveness. And then there was the replication problem. The trust, a lot of people tried to replicate the trust study, but most of them couldn't find the same results. That might be for a multitude of reasons. It could just be because they were using different cohorts of patients, the population was different, they were using different administration of oxytocin. But in general, people came towards a conclusion that it's more of a social salience hypothesis. And what that means is really perhaps oxytocin is making you more aware of the social cues around you, but how you react towards that social cue really depends on your personality, your environment, your genetic, your upbringing. And it doesn't specifically just produce positive effects. Right, so that is why I use oxytocin. So despite its shortcomings, it does and has shown, in fact, to increase positive behavior in animal models, and which is why I'm using it for my research. But the problem with giving oxytocin is that if you think of a conventional method of administrating a drug, you think of giving it by injection. So for example, if you give a subcutaneous injection, it goes into the fats of your tissue, and then it circulates to the blood and the rest of your body. But the problem is, if it wants to reach the brain, it encounters a problem called the blood-brain barrier. So your brain has multiple blood vessels, right? So the problem with it is that your brain is designed to keep things out. So, and that's where the blood-brain barrier comes into play. So only certain molecules, for example, it has to be smaller than a certain molecular weight, or it has to be fat soluble. So for example, glucose is something that is able to penetrate the blood-brain barrier from the blood vessels going into your cerebrospinal fluid, your brain fluid, essentially. So other molecules, for example, oxytocin is one of that would not really be able to bypass this easily. So a lot of animal studies actually use something called intracerebroventricular injection, which basically injects oxytocin directly into the brain. But obviously we can't do that in human patients because it's expensive and it's invasive and it's just not ideal in general. So we came out with another alternate method of administration that is quite popular. It's called intranasal delivery. So intranasal delivery, if you give like a nasal spray, for example, and you spray it up the nose, most of that drug would be swallowed and recirculated into your gut and into your blood system. And some of that might go into your brain, but obviously that would be a very, very little amount. Two main ways of how it might get into your brain is really through the nerves that are available in the back of your nasal cavity. So these are the olfactory nerve. So the olfactory nerve at the back would lead to your olfactory epithelium, olfactory bulb, which is the front bit of the brain and is responsible for smell. And via the trigeminal nerve, it goes towards your brainstem. So kind of like the bit right above your spinal cord. So once it reaches the top and the back of your brain, it then can diffuse to the rest of your brain. So yes, so the pros of intranasal delivery really is that it bypasses the blood-brain barrier issue that we mentioned earlier before. It directly penetrates the brain. And obviously it reduces side effects, because if you remember the earlier slides where I said that oxytocin had a lot of effects on like different parts of your body as well, it can reduce those side effects, because what we really want is just the social interaction, social behavior kind of effects in the brain. But then again, your nose is designed to keep things out, just like your brain, even though it's a bit more easier to access. So there is also an issue with, is there enough oxytocin to penetrate your brain? You still have to give quite a huge amount of oxytocin in order for you to see effects. So how can we improve intranasal delivery? Is really my research question. So what I have is, so my research question explores oxytocin versus a novel peptide of our lab. So we basically combine oxytocin with a different peptide, which is a carrier peptide essentially, and whether this carrier peptide can help transport oxytocin via the nose into the brain better than just oxytocin by itself. Yeah, so that is all of my research question. This is just a few pictures just to show you guys what my life is like outside the lab. This is a typical lab bench, so it's a bit messy, but you have all the appendals, pipettes and stuff like that to do experiments on. This is my lab mates around on the top here. This is someone passing his PhD viva and so celebrating them. And I do a lot of science communication bits on the side as well. So this was at British Pharmacological Society's fair, at one of the fairs, and this was at New Scientist a few years back, I think. And then just a shameless plug as well for my platform, 100 Scientists of Malaysia. Our aims really is to increase diversity of STEM, but visibility of our Malaysian scientists, inspire the public to engage and pursue STEM. We are on Facebook, Instagram, and Twitter. I think that is it for my presentation.
So we do have a few questions that we have collected online before this. And first of all, Sarah, so what you mentioned for your research is kind of like a way to boost oxytocin intranasally, so administering it externally, right? But is it possible to boost oxytocin naturally? Are there things you can go about during your day to try and get more oxytocin?
Yeah, so like I mentioned in the very beginning, so you can boost your oxytocin levels naturally by doing things that make you happy, really, by connecting with loved ones, by playing with your dog, by interaction with friends and families. That's why I think this kind of social isolation thing is hitting a lot of people very hard, because you're doing things that stop you from feeling happy again. But those things really, the social bonds that you have with like a pet or with your friends and family, those are really like important things in life.
Okay, definitely. Right, so we are getting a few more questions in the chat. I'm gonna ask some more general questions first about oxytocin. Is oxytocin genetically inherited, so the levels of oxytocin, or is it more of an environment or upbringing that plays a role in your normal levels of oxytocin? So is it a sort of a nature versus nurture debate, like which maybe has more of a role in your levels of oxytocin?
Yeah, so I think the thing with oxytocin is that a lot of research has been more emphasized on giving oxytocin and seeing what happens, because it's something that was already naturally produced. So people kind of ignore the bits before. And it's quite hard to say, do a longitudinal study and measure oxytocin across the lifetime of a baby growing in this environment versus a baby growing in that environment for 20 years. So I think they have been shown to have genetic, for example, SMP variability in the oxytocin receptor gene. So these are basically, you know, like your genes are made, your base DNA base sequence are ATGC. So
Yeah.
the SNPs are basically substitution of those base nucleotides. And then some variations have been linked to increased susceptibility towards the positive, negative cognitive symptoms of schizophrenia. But there is definitely more research to be done on that sort of things, that side of things, which I think has been a bit neglected.
Yeah, definitely.
Yeah.
I think it's very hard to answer the gene question for a lot of these things, for sure. All right. So how useful is oxytocin in improving social cognitive deficits, such as in autism spectrum disorder? So you did talk a little bit about that when it comes to schizophrenia, but what about social disorders like autism?
Yeah. So I think I can't really speak a lot for autism because my research lab focuses on schizophrenia rodent models, but obviously their symptoms kind of overlap, the increased social withdrawal, reluctance to social interact, cognitive wise. I think there have been studies in humans that shown that oxytocin does increase eye tracking. So I think eye tracking is like a way to measure attention, that sort of stuff. So I think like psychology students would be more able to explain what eye tracking really means, but it does show that it improves a bit of like eye contact, attention, gaze in autism spectrum disorders.
Okay, right, that's cool. What is your, so this is more of an ethical question actually. What is your opinion on the ethics associated with administering hormones like oxytocin, especially to humans? Do you think in the future, if we have more medication based around sort of hormonal treatment like this, would it be quite controversial?
So I think with oxytocin in general, it's quite different from other hormones, for example, growth hormones, which obviously would have quite a negative impact if you give it too much. Oxytocin in this sense doesn't really face that sort of problem. So I guess where the ethics come in might be, say, that trust study that I mentioned. You might get people being like, well, I mean, I was under the influence of oxytocin, that's why I decided to trust this person and give my money all away. So that maybe is something that people might play with, but I don't see a huge ethics problem with oxytocin as a hormone treatment compared to a lot of other hormonal treatment.
Yeah, just a follow up on that. I think you mentioned that there were some studies showing the negative effects of oxytocin, whereby some people had actually more adverse behavior after being administered oxytocin. So in the future, would maybe medics or scientists have to carefully monitor a person's background in order to decide whether to administer this kind of treatment to them?
Yeah, I think that's definitely something that we have to look into, because in animal models, it's a lot more easy to control their background. You can make sure everything is the same, right? But when you go to humans and bigger clinical trials where you get different kind of people across the world administering oxytocin, for example, maybe that is one of the reasons why we haven't seen good replication of oxytocin studies, because everyone is different. So I think understanding more molecularly wise, like what does oxytocin do? How does it exert its effects, instead of just looking at the really at the right end of the spectrum, like the behavior effects, but going a step back and seeing how does it exert its effects in different kind of populations, different kind of traits, people with different traits, and then going from there. That might be more of a personalized thing in the future.
Personalized medicine, yeah. That's really cool. This is a cute question. Do insects have oxytocin too? Can they feel love?
Okay, I can't remember this off the top of my head, but oxytocin has kind of like a history to it. So it really started with a jaw vertebrate mammal, and then insects and birds do have oxytocin, but it's not called oxytocin, it's a variant of oxytocin. So I have to look that one up because I have it in my lit review somewhere, but I don't want to say the wrong thing. But they have a similar thing to oxytocin.
Okay. A follow-up on that, how translatable is sort of rodent research to human research when it comes to things like oxytocin? Because you mentioned it's a bit different in insects and birds, but how different is it in rats compared to humans?
So rodents, rats and mice are one of the more commonly used ones in research, in neuroscience research especially, because their brains, the anatomy of the brain is quite similar to a human. And then they do rats as well, they are very social interactive. They are quite smart in learning things. So that's why we use them in a lot of cognition and memory-based tasks. Translatable wise, obviously it's not a perfect model for diseases such as schizophrenia, autism. There's so many symptom domains to it, and you can't really show everything in one single rodent model. So for example, for schizophrenia, the PCP phencyclidine that I mentioned is a pharmacological model. So we induce that by giving a drug, essentially. But then there are a lot of different other models, for example, neurodevelopmental models where you give the mom rat, when she is pregnant, a toxin to kind of stimulate environmental stress. And then the pups, when she gives birth to the pups, you can isolate the pups so they grow up alone, or they grow up with their little mates. So that it's kind of like an environmental stress, neurodevelopmental stress. And so that kind of targets different aspects of the schizophrenia underlying etiology. And then there's also genetics models of her. So I think it's more of, by everyone publishing their bit of research, you kind of gather everything and look at the bigger picture and decide, piece everything together and figure things out from there. So there's no one question, there's no one answer fit one question kind of thing. But I guess in the long run, it is translatable in some sort. So as long as you do sound research and you go about your techniques properly and you have confidence in your results, I think that is what matters. And you're using the animals properly, of course.
Yeah, I think that issue with translation, it impacts many science fields, because we develop all our models, but we don't know how they actually perform in real life, right? When you're actually acting human lives. So that's always very interesting to talk about.
Yeah, but you think of like all the medicine that you have on the market, it all came from cell studies, then moving on to animal studies, safety studies, all that is done in animals. So it is translatable, but you can't. There are bits that will obviously be different in animals compared to humans.
There are steps to it, of course. Yeah. Was also wondering, has there already been intranasal sort of delivery of oxytocin based treatment in humans already, or is that far down the line?
It's currently clinical trials, but there has been intranasal formulation for other diseases. I can't for other peptides. I can't remember them off my head now, but there has been I think that was one recently, not recent, but a few months back. It's a drug for depression, if I'm not mistaken, but I need to look that up probably.
Okay. So do some people have less levels of oxytocin than others? So are some people born, with less levels?
Yeah. Again, with the born question, it brings us back to, it's quite hard to measure from like birth to grown up level. So I don't think there's been studies done on that. So what studies have done is maybe comparing schizophrenia patients with control patients. That in itself is controversial because some people found increased level of oxytocin, some find decreased level. So there's not really a consensus. They just have a consensus that it is impacted somehow, but how is it impacted? They don't really know. With age, again, in rats, they've been shown that oxytocin receptors is more available in some parts of the brain in a juvenile rat compared to adult rat. In humans, again, this has not really been done, but they did find that oxytocin receptors do kind of decrease as you age, which I guess will kind of fit well because as you grow older, your cognitive functions does decrease. So maybe oxytocin does play some sort of role, but maybe it might not be a main role as well, because obviously there's other neurotransmitters that are involved in these sort of stuff.
Right. We might move on to some more technical questions now. One of them is, do you think a sustained release formulation would help for delivering oxytocin intranasally?
So the thing with sustain, if you keep giving oxytocin, so one special thing
sorry, Sarah. Before, I should have said this. You could describe what that means. So sustained release formulation.
Okay. So if I'm not mistaken, sustained release formulation would basically be so that there would be a constant release of oxytocin. Yeah, constant release of oxytocin, essentially.
Okay.
Yeah. So with this, oxytocin is special in that sense because like a lot of other peptides, there is a negative feedback loop. So that means once there reaches a saturation of, you've got A levels of drug A, your brain will be alerted to that and be like, okay, we need to reduce and dampen the levels so it doesn't go overboard. But with oxytocin, it's a positive feedback, so it's the opposite. So for example, in labor and childbirth and breastfeeding, when you release oxytocin, it stimulates milk letdown, for example. Then your brain registers that, oh, there's this level of oxytocin. Let's create more oxytocin so that we can release more milk or help contract the muscles even more. So in that sense, I think you do have to be very careful when you talk about like sustained release formulation, because then the positive feedback might be a negative effect instead of a positive effect. And oxytocin is better in short bursts of release compared to like constantly giving it.
Right. So this harks back a bit to the ethics question in the beginning. It's more very technically, how do you minimize the sacrifice of your rats while still getting samples and answering your research questions?
Yeah. So when it comes to animal research, it's a very controversial subject, obviously. I myself don't like the thought of animal research, but I can see why it's needed. So I can speak for the UK, not for anywhere else. So in the UK, to do animal research, your PI, your boss of the lab essentially has to apply for a license with the Home Office. So, and that is quite a stringent process, and it contains a document that goes through every bits and pieces of like, we need backup of why you need to use this model, and it states everything that you can or cannot do to the model. For example, if you do a fear experiment and that involves shocking the rat feet, it doesn't harm them, it just gives them a short electrical shock. The license will stipulate like how many times you can shock the rats for and how long you can shock it for. So you can't just, oh, 5 shocks doesn't seem enough, maybe I'll try 7. You can't do that because your license will state that a maximum of 3 is all that you can do. And it also stimulates like how much you can do to your animal models in one day. And then there's also something called a power study. So you have to, basically the 3 R's, to reduce, reuse, and recycle. You have to do power calculations and stats to calculate how many animals would be sufficient in one treatment group to show stats difference, significant difference, so that you don't use too many. By the same time, you don't use too little, because when you use too little, then you have to up the numbers, and that would involve waste. And then another thing I'll point out about animal research is that the animals are very well cared for. Like we have facilities and we have members of their staff members that take care of these rats, of these animal models, not just rats, so that they are well fed, well watered, well taken care of. Really, they have toys to play with for enrichment and that sort of stuff. And there's also a vet that always comes every week to make sure like everything is okay and in order.
So on to the more sort of in the future type questions. What is the future of neurobiology-related careers in Malaysia?
So I don't think I'm the best person to talk on how the neuroscience field is like in Malaysia, because I've not experienced it. There are labs that do neuroscience research. For example, I know a friend at who is doing mushroom extracts on neuro regeneration, neuroprotection kind of stuff. So does definitely have some sort of neuroscience department. Another famous one would be BRIMS, Brain Research Institution at Monash. So they do a lot of brain research, obviously. So that's something that you could Google and look up the list of researchers in there, and then go from there.
Okay. The next one is quite a cool question. What is your dream research question? And do you think your current path will lead you to finally answering that dream research question in the future?
Thanks, Yao Hua, for that question. My dream research. So I definitely want to stay in neuroscience, definitely into neurodevelopmental disorders, psychiatric diseases. So I'm interested in anything to do with really like depression, schizophrenia, autism, that sort of diseases. What goes wrong, the treatment. So pharmacology would be like the drugs that used to treat them, how does the drug works. So I'm interested in that field. would my current research lead me to my future dream research path? Maybe, I'm not quite sure. Like, I'm interested in oxytocin still, but will I continue in oxytocin? I'm not sure. But I do find the blood-brain barrier aspect of it quite interesting, like improving brain penetration of certain drugs. That is something I find interesting, but I think I have a lot of interest. So it really just, whatever feels interesting to me that lies in that kind of domain, I will go towards, and then maybe future on, I'll get a sense of like, what exactly is my research question that I want to focus on.
Definitely. So on to a few more questions. So there are some additional, more oxytocin related questions, but I'm just going to finish off the more sort of future-based questions first. You did answer this a little bit in your last answer, but what are your career aspirations after your PhD? And to expand on that, do you think you'll still be doing science communication alongside your sort of research postdoc career, or will you focus more on one or the other?
It's definitely a mix of both. So I do want to stay in academia. So postdoc would be something I'm interested in after my PhD. So that's the academia real job kind of side, paying job side to it. But science communication is something that I still feel very strongly about, especially I think during my education in the UK and seeing like how strong science communication is done here really makes me think like this is something that I want to emulate back in Malaysia, which is why 100 SOM came up with Josh and Anthony, who founded this together. So yes, I'll be doing both at the same time. We are still planning to keep 100 scientists of Malaysia. A lot of people ask like, oh, after your 100 scientists, what's going to happen? So we are going to continue featuring, maybe not as much as strong as a pace as we are now. Like I think we do 2 to 3 a week. So we'll probably cut down on the frequency, but we are looking into a lot of more physical events. So for example, we are looking at providing research internships for Form 3 to Form 6 A-level students before they enter university, to kind of give them a taste of what it's like to do research in a lab, and also so that they can explore the different views that they might be interested in, and then be like, oh, do I want to do neuroscience, or do I want to do biochem, or do I want to do genetics? Is this what I really like? Once they get a taste of the research lab, so that might help them choose their degree when it comes to university. So
no, that's such a good idea, because I think if I were Form 3 or A-levels, I would super appreciate being able to do an internship first, deciding what I want to do for the rest of my life, right? So that's really cool. Yeah. So jumping off of that, can you share your experience a little bit about co-founding 100 Scientists of Malaysia? So how did that actually start? What's the story behind that?
So Josh came up with the idea, really, the initial idea of forming this platform where we tell the story of different Malaysian scientists. I think he told this to me and Anthony. So we are housemates in Nottingham, because we are all PhD students at Nottingham. So we agreed to the idea, and then we came together, and it took a few months of hashing out how to best deliver these sort of features, because there's so many platforms out there that just type really long paragraphs and posting it every other day, and then just be like, yeah, here you go, like just giving you really lots of information. But because this has to do with science, we know that science can be quite intimidating with all its jargon and whatnot. I myself might be fought with that as well. So we had to deliver it in quite a fun way, which is why for our Instagram we do like the photo cards version. So it's a really TLDR version, and you can really easily read through. And then for our stories as well, half of it would be about the science, but then the other half we would focus on the story, the message that we really want these scientists to send out to inspire people. And I think we've got there because people have been giving quite a lot of positive response. We did get a lot of support in the earlier months when we were just starting out as well. So co-founding 100 SOM, I think we didn't think that it would be as popular as it is now. We were hoping it would have some effect, obviously, because we dedicated a lot of time to it, but we didn't expect it to be this well received by the public. So that's really nice. And it showed that really there is a gap in Malaysia, in the Malaysian society for this kind of things.
Yeah, for sure. Because I think many Malaysians are very interested in research and what kind of science is happening, but there's not always a platform out there that communicates that science so effectively, right? So what you guys are doing is really cool, making science very digestible for everyone. And I really commend you for that. Right, so we do have this additional question that, again, sorry, we're looping back into oxytocin because this is a question that came a bit later. So when it comes to this intranasal administration of oxytocin, does the treatment only provide temporary relief of the symptoms? Is the idea to self-administer when the symptoms flare up, or is it sort of a constant treatment that you take maybe once a day?
Yeah, again, this is a study that has to be, again, it comes back to time. So a lot of animal model study, they always check for behavior effects or anything that they can, so they always check for behavior effects within hours of after administering oxytocin. So that there really isn't much studies out there that say administer one dose of oxytocin and then check on it a few months later, or giving oxytocin for months and then checking the effects. It's always quite 2 weeks, 4 weeks. I think the maximum is always usually around a month. I think this is something, so temporary relief, I think, yes, because you are not treating the root of the problem. You're not treating like the receptor problem or any other problem that may be the root of, saying, causing schizophrenia or autism. You really are putting a band-aid on something and covering for the symptoms when it flares up, if that makes sense. So really putting a band-aid and not going to the root of the problem. So this would not treat, it would just treat the symptoms but not the disease.
For sure. Would this be kind of akin to, asthma inhalers, where when you're
I would say so.
Yeah. Do we need to limit ourselves to a certain research focus, which I think is what a lot of PhD students tend to do, I think, when you're in a PhD sort of level degree?
So I think when I was in third year of undergrad, my final year, and I knew I wanted to do a PhD, I went to my personal tutor and I was like, I have so many things that I'm interested in. What do I choose? Like, I'm scared of choosing the wrong one. And then what he told me was that it's good that you have multiple interests, because then you have multiple choices. It's better than having no choices and nothing at all. So I think instead of being like, oh, I have too many choices, embrace the choices that you have. Go with your gut feeling. And obviously, if you can, try immerse yourself in related experience, or if you're interested in this experience, try and find experiences related to that and see if you actually enjoy it. Do internships if you can, obviously, lab internship, that really helps a lot. It tells you whether you're ready for research, but limiting yourself to certain research focus, I think it comes as you go along your research, your career path. I think the older you go, the more knowledge you gain, the more interested you might be in a certain research area. So maybe don't close yourself off entirely to anything. Keep an open mind, but then let it kind of lead you along further in life.
Great answer.
Ride the waves. Yeah.
Yeah. Okay, cool. Right, so we're reaching the end already. Thank you so much, everyone, for joining us for this very, very illuminating session of Research Insider. We have a poll here for people to fill up, because we do want to hear your feedback about how we're doing with these sessions, because we really like putting them on, but we just want to know if we could be improving anything. And yes, thank you. For more updates regarding future Research Insider sessions, please like our Facebook page, MBIOS and 100 Scientists of Malaysia. And for those who joined late or missed our science chat, you can watch the edited video on our MBIOS YouTube page. And I just want to say a huge, huge thank you to Sara once again for joining us for this very, very cool session. And thank you everyone who managed to join as well. Right.
Thanks for joining, everyone.
Yeah. Okay, thank you. See you next week, everyone.
Yeah. Bye.